My name is Dorit, and I'm a postdoc at Stuart Lipton's lab at Scripps Research, and we use human-induced pluripotent stem cells to study neurodegeneration and neurodevelopment. And today I'll present our model of autism spectrum disorder and how we use that to interrogate neuronal abnormalities. So MEF2C haploinsufficiency syndrome is a form of autism, and autism spectrum disorder occurs in 1 in 54 live births according to most recent estimates. There's an association between MEF2C mutations and MEF2C haploinsufficiency syndrome, and I'll talk about MEF2C in more detail soon. MEF2C haploinsufficiency syndrome is characterized by severe intellectual disability, epilepsy, stereotypic movements, minor dysmorphisms, and brain abnormalities in addition to autism. And in the last 10 years or so, there's greater interest in MEF2C haploinsufficiency now that many children diagnosed with autism go through genetic testing. So here you can see the association for MEF2C Facebook group, which is mostly European group. And this is the MEF2C.org website where these families and patients can get to know each other and support each other. And that's part of the way a lot of them reach our group. So we've met many of those patients as well. MEF2C is also reportedly a co-regulator of a lot of gene networks involved in other forms of autism. And this is work from Dan Geschwin's lab at UCLA, where he showed that MEF2C can be a hub for several gene networks that they found in both genetic and sporadic cases of autism. So MEF2C is part of the MEF2 family of transcription factors, which are important for development, differentiation, and organogenesis. And in the brain, MEF2C drives the expression of neuronally restricted genes. And it's important for neurogenesis, neuronal survival, and it regulates synaptic plasticity. Our group discovered MEF2C originally and have shown that brain-specific knockout of MEF2C in the stem cell stage results in electrophysiological and behavioral deficits reminiscent of Rett syndrome. And this was before MEF2C haploinsufficiency was described. So once MEF2C haploinsufficiency syndrome was described, we knew that this phenotype had a physiological relevance. More recently, we've shown that MEF2C het mice exhibit decreased neurogenesis, enhanced neuronal apoptosis, and increased E to I, or excitatory to inhibitory neurotransmission. And so our work now is to try to use human-induced pluripotent stem cells to study this disease. And using the system, we obtain skin biopsies from children with MEF2C haploinsufficiency syndrome. We isolate primary fibroblasts. We reprogram them to human-induced pluripotent stem cells using the 4 Yamanaka factors to transform fibroblasts or de-differentiate them to iPSCs. And finally, we differentiate these iPSCs into neurons in a 2D culture or 3D organoids or mini brains. So to be more specific, when we differentiate the cells to neurons, we go through a neural progenitor stage using the dual SMAD inhibition protocol, which generate rosettes and then neural progenitor cells, and finally neurons. And this differentiation protocol contains both excitatory and inhibitory neurons. We also differentiate cells to 3D organoids using the Pasca protocol from Stanford. And this protocol yields organoids that have similar structures to the brain with what looks like ventricles and some layering markers. And in this study, we have 4 control lines and 4 patient lines. And I'll just draw your attention to patient 2, which is an isogenic line using CRISPR-Cas9 to control 1. So this is an isogenic pair. The other ones are from patients, and we tried as best as we can to have age and sex matched. The first thing we looked at is we wanted to see the network activity of these iPSC-derived neurons from controls in patients. So to do this, we used calcium imaging as a correlation of electrical activity. So I'll show you a video first of normal iPSC-derived neurons, what looks like normal network activity. Some cells have more or stronger calcium signals, and you can see the propagation from cell to cell. When we move to patient line, we see what looks like a very synchronized hyperexcitability. This is reminiscent of a seizure, which, as I mentioned, many of MEF2C haploinsufficiency syndrome patients do have seizures. And so when we saw this striking effect, we wanted to really understand what could be the mechanism for this. So we looked at two different time points, early development and later activation of neurons. The first thing we did was to look at MEF2C activity using reporter gene assay by luciferase, and we looked at cells as they terminally differentiate from neural progenitors to neurons. So day 0 is NPCs, and then they differentiate to neurons. And at about day 5, we see a strong increase in MEF2C activity, which is not the case for the patients, which is well expected since they do have only one normal copy of MEF2C. Next, we looked at the ability of these progenitors to generate neurons and astrocytes. We labeled with GFAP for astrocytes and MAP2 for neurons. And if this is the control line, the patients had much higher GFAP signal and less MAP2, suggesting more gliogenesis and less neurogenesis. We then performed chromatin immunoprecipitation sequencing to find MEF2 targets, and we focused specifically on microRNAs that had lower expression in all of the patients. And when we inhibited these microRNAs in normal cells, we found that recapitulated the phenotypes of patient lines, meaning fewer neurons and more astrocytes. So our working model is that in MEF2C haploinsufficiency, since there's less MEF2C activity, fewer microRNA, and more gliogenesis. This is what organoids look like. And we also found that the organoids from patient lines were smaller and had fewer neurons and more astrocytes as well. And this is the quantification. And finally, we wanted to address the hyperexcitability phenotype. So we looked at later stage at excitatory and inhibitory synapses. MEF2C is also reported to be a transcription regulator of parvalbumin positive inhibitory neurons. So we stained here for VGAT and VGLUT1 for inhibitory and excitatory synapses, and found that the patients have less inhibitory synapses, which could explain the hyperexcitability. And so to conclude, we suggest that MEF2C, in MEF2C haploinsufficiency syndrome, there's lower activity of MEF2C, less microRNAs, and then higher gliogenesis. And among the remaining neurons, we have fewer inhibitory neurons, leading to hyperexcitability, higher calcium signals, and hyperactivation. And with that, I would like to thank my mentor, Dr. Stuart Lipton, my collaborator on this project, Dr. Swagata Gatak, who performed a lot of electrophysiology. And I encourage everyone to look at our recent bioRxiv paper to see a lot more of the electrophysiology characterization, and to our collaborators, Nick Shork and Dan Geschwind, for helping with bioinformatics, and Michael Rosenfeld for the ChIP-seq assistance. And thank you for the funding and for you for listening.
Thank you very much, Dorit, for this really good talk. I would like to address our panelists and ask if they have any questions for you. We also have a question from the audience. Uri Weil asks, can you rescue the patient lines?
Yes, so we currently have two strategies. One is to activate the normal copy of MEF2C, and that could help at an earlier stage of development to prevent the aberrant gliogenesis. And we also have a drug that prevents hyperexcitability at the later stage, which is more a phenotypic type of treatment. And we were able to use both of these to rescue the phenotypes.
So from Asaf Citrin from the audience, there is any correlation now to any breakthrough of medicine to autism?
I'm not sure. I can say that this is correlated with other forms of autism, specifically Rett syndrome, MECP2, and MEF2C, or MEF2C is upstream of MECP2. And I think some of the more recent treatments for epilepsy and things like that work well with these patients. But I think right now I can't say that there's any real breakthrough that helps these kids, as far as I know, unfortunately.